We're going to now sort of listen to a couple of life science highlights to remind people of some of the really exciting things we might be able to do and deliver as part of the program. So each speaker is going to speak for around 12 minutes, and then there's a few minutes for a Q&A just before lunch. So to kick us off will be Natalie Cohen. Go ahead, Natalie.
Thank you. Hi, everyone. I'm excited to be here with you today. I'm going to speak to theme 1 and walk you through a couple example data sets where we examined shifts in microeukaryote taxonomy and function across spatial dimensions. I'm going to start off with some background. Microeukaryotes, also known as protists, play key roles in ocean biogeochemistry. Photosynthetic protists, fixed carbon, heterotrophic protists are important grazers, and protists contribute to the vertical movement of carbon in the water column. Phytoplankton are confronted with micronutrients limitation and co-limitation across a large region of the ocean. Of course, in the equatorial Pacific and the Southern Ocean, we have primary iron limitation. And then in contrast, in the North Atlantic, iron is introduced through Saharan dust. And here, primary nitrogen limitation occurs, but phosphorus and iron co-limitation can occur. Microeukaryote communities are incredibly diverse. We've made progress over the past few decades in understanding their diversity, but this is still an open question in terms of their diversity and functional roles in large regions of the open ocean, and especially in the deep ocean. As molecular ecologists, many of us are interested in their transcripts and proteins because these can be sensitive to environmental change and help us understand how these organisms are perceiving their environment. Nutrient biomarkers in particular can be valuable. These can be examined in either the transcript or protein pools and can give us a sense of where microorganisms are experiencing nutrient stress or limitation and how this changes across spatial dimensions. Ultimately, their physiology is important for us to understand because it can help us figure out how they're contributing to the efficiency of the biological carbon pump. I'm going to very briefly outline 2 different gradients, the first one being in the Central Pacific. In this transect, we started off in oligotrophic low nitrate waters and then sailed into the equatorial upwelling region, which is characteristically iron limited and nitrate is elevated. I'll then shift gears and introduce a gradient in the Western North Atlantic where we sampled along an offshore coastal transect. The main questions for this first Central Pacific gradient were which microeukaryotes were dominant in this region and how did it change both laterally and with depth? And were they sensitive to these shifts in nutrient resources? So for this cruise, as I mentioned, we sampled both laterally and with depth into the mesopelagic. We used a combined omics and geochemical approach. The omics was metaproteomics and metatranscriptomics. Here is a section view of that transect. We have our northern end here on the right and our southern end on the left, and I'm showing you nitrate concentrations down to 800 meters. We see that nitrate was quite low in those oligotrophic gyres and elevated at the equator and a bit south where the upwelling was happening. In black, these are all sites and depths where we collected biomass using McLean pumps. They are battery operated, attached to the winch line, and they filtered hundreds of liters of seawater. So we used this material to perform our multi-omics analysis. So we sampled across this 4,600 kilometer section. Using our bioinformatic workflow, we ended up with 500,000 annotated open reading frames or predicted proteins. And through mass spectrometry metaproteomics, we identified just under 10,000 proteins. So one of our key questions here was which microeukaryotes were present and how, if at all, did they shift across these dimensions. Showing you 18S rRNA amplicons on the left, transcripts on the center, and proteins on the right. We are looking at all of our different sites and depths. So up top would be samples collected at our more northern sites, and down at the bottom would be samples collected from the southern end of that transect. And different depths are embedded here as well. And this is all relative abundance, so it's scaled to 100%. The colors are different microeukaryote supergroups. Importantly, this was biomass collected from the 3 to 51 micron size fraction. So it absolutely contains eukaryotes. It contains a lot of bacteria as well that I'm not showing here. Our annotation database was PhyloDB, which was created by Andy Allen's lab. So what we see here is that dinoflagellates, which are this purple group, were relatively abundant throughout the transect into the mesopelagic and across that lateral gradient. Diatoms are commonly thought of as organisms that do well in upwelling environments where nitrate is available, but here we learn that dinoflagellates are also ecologically successful across a large region of the ocean, and these offshore dinoflagellates are not well understood. We next asked how these dinoflagellates were metabolically responding to those changes in nitrate and iron. Starting off here with nitrate, showing a zoomed in version of the figure before where we're looking into about 400 meters into the water column. And we see low nitrate in those oligotrophic gyres and that signature of upwelling with the elevated nitrate. We looked at a few different nitrogen stress biomarkers. The one that I'm showing here is a nitrate transporter. It's been commonly observed to be upregulated under nitrogen starvation conditions in eukaryotic phytoplankton in culture. Here in these dinoflagellates, they more highly expressed this nitrate transporter in those oligotrophic gyres, and they expressed less of it in that upwelling zone. Iron distributions generally look very different. Iron is low throughout the surface, but we saw an interesting feature south of the equator where the ferrocline deepened and that low iron water extended deeper into the water column. We examined a number of different iron stress biomarkers. The one I'm showing here is iron starvation-induced protein 2A, also known as phytotransferrin. It's an iron transporter commonly expressed under low iron conditions. And we see its signature higher in the southern end of that transect, presumably where these organisms have nitrate and they're growing into iron limitation. We conclude here that these offshore dinoflagellates are sensitive to changes in nitrate and iron concentrations and will upregulate transporters as needed. All right, I'm going to move on and introduce the Western North Atlantic. In this data set, there are many different questions that we set out to address. One that I'll talk about here today is how the microeukaryote community changed throughout the entire water column. In the previous data set, we were mainly looking at the surface and mesopelagic, but here we have data from the bottom of the ocean. We were also interested in whether there were shifts in nutrient physiology in this very different nutrient regime compared to the last data set, but I won't have time to talk about that today. This cruise started off in the Sargasso Sea at the Bermuda Atlantic Time Series Station, also known as BATS. We then sailed into coastal waters that received subpolar influence from up north. We again performed a combined metaproteomics and metatranscriptomics analysis. Here is a section view of that transect. Here we can see Bermuda, and we ended up sampling along a continental slope. At one of our sites in particular, we were able to retrieve biomass from the bottom. We captured a number of different features. The one I'm showing here is turbidity, also known as beam transmission. And we see a nepheloid layer just along that continental slope, which is a region of suspended sedimentary material. The way that we collected biomass was with Clio, which many of us saw downstairs. Clio was designed by Max Sato, Mike Takuba, and Chip Breyer for in-situ microbial biomass collection. It has these cartridges that can be programmed independently to filter water from different depths. It's completely untethered from the ship, so the ship is able to carry out other operations while this is happening. So using Clio, we were able to filter on average 2,300 liters of seawater per dive. Each dive took about 13 hours, and we completed this section, this 1,000-kilometer section, in 11 days. The data that I'm going to show you for this data set was collected on the 0.2 to 51 micron fraction, so it includes both eukaryotes and bacteria, but I'm just going to focus on the eukaryotes for right now. This was the largest sequence project that I've worked on to date. We teamed up with Harriet Alexander, who's here, and her student Ariana Krinos, who's also here, to assemble and annotate these large metatranscriptomes. We ended up using the Eurythmic workflow and the ukulele tool that Ariana put together. We ended up with 7 million annotated open reading frames and about 77,000 proteins through mass spectrometry. The database that I used was a combined database using references from PhyloDB, RefSeq, and UkeProt. Importantly, we found that there were differences in your biological interpretations depending on which database you used, so we ended up combining them. One of our main questions here was what was the microeukaryote community along this gradient, laterally and with depth. Here again, we're looking at relative abundance going from 0 to 100%. We have our sites closer to Bermuda at the top, sites closer to the coast on the bottom. And the grayscale bar is showing you depth with our deeper samples in darker gray and black. So a couple features I'll point out. We had haptophytes, dinoflagellates, and stromatopiles, which include diatoms in the surface. We saw a transition in the mesopelagic to heterotrophic protists like Picozoa, radiolarians, foraminifera, and Discoba. And then at our site where we were able to send Cleodon to the bottom, we saw the emergence of fungal proteins. So we found these shifts in protistan communities vertically with depth. And I'm just going to quickly highlight that these fungi were detected within that nephloid layer. Here I'm showing fungal proteins on the left and then beam transmission in the center. What we know about this nephloid layer is that it's associated with lithogenic material, but also a slight enrichment of organics. Particulate metals are elevated in this region, and fungi are important consumers of organics throughout the water column. So it's unclear if fungi are just generally abundant in the bathypelagic or if they were directly interacting with this nephloid layer. And I'm almost done. To wrap up, both Clio and McLean pumps enabled high-resolution omic sampling across spatial dimensions. Multi-omics was valuable in reconstructing microbial community dynamics throughout the water column. And those biomarkers of nutrient stress can be valuable for tracking changes in physiology across gradients. There were lots of lessons learned that I'm excited to talk about later on. And thank you to funding sources, co-authors, and especially Matt. Thank you. Thank you.
Okay, we've got time for one or two questions. Yeah, can you mute yourself?
Oh, yeah, yes.
One here and then there's one over there. Okay.
Hi, thanks for your talk. I'm Nolwenn Lemaitre from Toulouse in France. I have a naive question. So about the first part of your talk, you spoke about iron starvation. Mm-hmm. How did you identify the transcripts that you had to look at?
Yeah. So we depend on annotation databases. KEGG is a really useful one that's populated with known proteins from model organisms. But there's also PFAM that looks at conserved protein domains. So we rely on annotation databases, but they're missing a lot of information. So there's a whole lot of proteins that aren't characterized that we're not able to see in that functional analysis.
One more just over here.
Yes.
Yeah. Thank you, Natalie. For the second part of your talk, the Atlantic transect, you showed this nice color-coded protein distribution.
Yeah.
Exactly. So if I look at it, I think it's amazing how similar these patterns actually are. If you look at it at a holistic with holistic glasses. For example, gray. What is the gray part, by the way?
Gray were proteins that we couldn't confidently annotate to a supergroup level. They were too similar across diverse organisms.
I see.
For example, it could be a pretty good match to both a dinoflagellate and a haptophyte.
I guess my question is, so what is it? Is it like what is the statistical threshold for you to say I see a difference here from one depth to the other?
I mean, that's a good point. So just what I'm showing you here is observational, like looking at the relative abundance. We see that consistently in surface waters, we see this like even nice diversity of these different groups. And then consistently, when we go into the mesopelagic, we see this more diverse community appear. I do have PCAs that I can show you later on, which show that depth is the most important factor in differentiating these communities.
Thank you.
thank Natalie again.
Oh,
Thank you.
thank you.
Okay, so the next speaker, just while we set up the slides before lunch, is Paul Fremont, who's going to talk to us about global scale climate genomics and how it describes something. The modulation of the plankton composition and...
A long title. So.
Yeah, you explain it yourself, Paul.
Okay. So thank you for giving me the opportunity to talk here. So I'm Paul Fremont. I worked previously at Genoscope on this project on the Tara Ocean Genomics database. And I'm now in the University of Maryland working on quantitative viral dynamics of ocean viruses and their hosts. So we're talking about plankton diversity in the ocean. So we know that marine plankton are key players in the ocean ecosystem. And so just a reminder, so that they are defined as a set of organisms that are passively transported along currents. And so they form these large communities and complex communities and interact with each other. And so they encompass a large variety of sizes, trophic modes, shapes, and biological functions. And of course, as we know, they play really important ecological roles. So they are at the base of the food chain with the photosynthetic bacteria and photosynthetic eukaryotes that are the primary producer in the ocean. They participate then to carbon export through the biological carbon pump, and of course they are involved in many elemental biogeochemical cycles. And so from 2009 to 2013, there was this expedition, Tara Ocean, that made this large-scale sampling of the ocean throughout a 3 or 4 year global expedition, and with genomic sequencing of all the different organisms of plankton. So first there is the expedition, then genomic sequencing, and then the science. And so, as you know, there are many papers that came out of this expedition. Well, I put it's a little biased towards the one to which I participated, but and so there are many more. So this was the empirical approach to size fractionate as we know that there are different size fraction organisms. Different organisms of different size. So we have to size fractionate them so in different abundance. So we have to sample with different size fraction. And so we have a consistent DNA and RNA sequencing of each size fraction. So with the metabarcoding that characterizes the taxonomy. But here I focus more on metagenomic and metatranscriptomic, which is shotgun sequencing of the community, but biased toward the most abundant organism, meaning that we cannot sequence the whole community and the rare organism with those methods yet. So those communities have different ecological niches, and they thrive in many environments. And here is just like kind of a simplified illustrative scheme of what we know of the different niches of plankton, and with different role in the fishing grounds, for instance, and also in the carbon export, and based on whether they are in nutrient-rich and cold or hot environments. And there is the problem of climate change. We have, of course, a temperature increase with so a lot of consequences on the environment, with increase in frequency, strength of marine heat waves, new mean local extreme, pH decrease, etc. I'm not going to read all of them. But climate models, they can project changes in the ocean biosphere, but with high uncertainties and low resolution, basically, on the biology. So we have we have a lot. We don't characterize well their displacements, how they participate to carbon export, to net primary productivity, how the trophic change might be affected, also changing in their phenology and their adaptability to climate change. And so this is the result from a Taro Ocean expedition. We compared metagenomes across all stations, and here we see the biogeographic patterns of one size fraction, so enriched in eukaryote protists, and we were amazed by this partitioning of the ocean into what we called metagenomic provinces. And so we have this for 6 size fractions, and so we extrapolated those biogeographies for the 6 different size fractions. So here, is there a laser? No. So on the bottom right, the viruses, and towards bigger organisms at the top left. And you see that, for instance, small metazoans at more simple latitudinal biogeography, whereas small organisms have more complex patchy biogeography that fits more big oceanographic structures like the oligotrophic gyres, the equatorial upwelling that we see in the Pacific, for instance. And we are able also to assemble into metagenome assembled genomes, those metagenomes. And so we are able to characterize the organisms that define those provinces. So mainly here are the large size fractions with copepods. But you see, for instance, in the size fraction 520, there are also a few. There is like pseudonychia that characterize, so a diatom that characterize the equatorial community of this size fraction. And so then we did projection with climate change by the end of the century, climate models, so CMIP5 intercomparison models. And we project our biogeography, and we project a large recomposition of our communities, and we associated also this with carbon export data, and so we could project a decrease of 4% on average of POC export flux at 100 meters. And in the second analysis, I wanted to I'm working on right now is to see actually what distinguish this province functionally. So okay, it's good. We know that there are some organisms that are here and that are different across the different parts of the ocean. But what actually distinguish them in terms of their functioning, and if they might have some impact on the biogeochemical cycle. And so I took here we take the example of the North Atlantic Ocean compared to the Arctic Ocean. And here you see the transport time between the station and the main currents, and you see that actually there is a strong environmental gradient that the communities are encountering with what we call the polar front. So we wonder how organisms are responding to this front. So sorry. And so here we see, for instance, that in large eukaryotic group, and so for different metabolisms, we have actually some changes across this gradient. So for instance, in mammillal and diatoms, the main photosynthetic transport chain was turned down. There was downregulated. Thank you. In the Arctic, whereas it was not the case for the Phaeocystis. Also, we see that the light inverse complex are still maintained with high expression in diatoms and mammillal in the Arctic. One feature also that we see is like different expression of the nutrient transporters. So, for instance, in the mammillal, they have high expression of phosphate and nitrate transporter at the almost in the whole sampling zone, but lower in the summer Arctic, so the last stations. Whereas diatoms and the photosynthesis, they had more expression of ammonium transporter, which was more upregulated in those organisms. So which might be linked to their to the small size, for instance, of Mammillaria. So I don't have time to describe all of this. And one other analysis that we did is that we showed that there is a common set of functions that are also responding to the temperature gradients when the organisms are entering the Arctic. So we reconstructed a metagenome-based transcriptome, so MGT, and we do the correlation of the gene expression with temperature, and we see that there is a high, there are a lot of functions that are, for instance, related to stress and cold, energy consumption, and also a lot of genes related to gene expression regulation. So from signal pathway to post-translational modification. And we see that this response is common across the different groups of algae. So diatoms, photosynthesis, Batycoccus, and Pelagophyceae. So we see this convergence in the expression. So to summarize, we have those global transkingdom biogeographies of plankton with the signature genome. So we described who is where, and we also projected the effects of climate change. And there might be some impact on biogeochemical cycles with 4% decrease in carbon export flux, for instance. But what I wanted to stress out today is that the sole presence of organisms, of course, is not sufficient to describe the way they function in the ecosystem. And so for this, I took the example of the Arctic Ocean polar front, and we see that some species are crossing the polar front, and that they change their physiology as they cross the polar front, and also the autotroph metabolisms are also different across the different groups of eukaryotes between the Arctic Ocean and the North Atlantic Ocean. But those eukaryotes, they also shared like a similar acclimation response to cold while crossing the polar front. And yes, one important thing is that, for instance, the gene expression of photosynthetic genes didn't correlate, for instance, with biological rates like net primary production, so which I think is a big challenge for the biogeoscape community.
Thank you, Paul. I think we've got time for one question, so we can get to lunch. Anyone have a question? Paul?
Okay, this might be a really bad question, but all the sequencing was done on size fractions that were separated by filters, right?
Yes.
How much, this is a little connects to Natalie's talk where you noted all of the bacterial sequences you found in the 3-micron fraction. For you then, how often or how regularly did you find nucleic acid information from groups that were not what you expected in that size class? Does that make sense? I mean, was that really common, like Natalie saw as well?
Yeah, like as I showed an example in the, there are a few examples of this. In the big size fraction, there is the 180 to 2000. There are some photosynthesis that are in the Antarctic, in the few Antarctic stations. That are actually they probably form chains, so they are in this size fraction. And in the 520 micrometer, also I gave the example is that you have a mix of large eukaryotes, well, relatively large eukaryotes, and some arthropods.
Thank you, Paul. Let's thank Paul again.
Thank you.
Can I also just announce that Mike Jakuba, the Clio engineer, is going to be hanging out by the vehicle downstairs during lunch if people have questions and want to see it. Also, he'll be up for a poster session later today. And then with that, we go to lunch, right? All right. Thanks, everyone.
Thank you, everyone.